Hello, everyone. It's Mr. Vallejo. Welcome to science class. Today, we'll be taking a look at the intertidal zone. So let's go ahead and share the screen and go to our PowerPoint for today and take a look at the intertidal zone. Here we go. Okay. So this is in reference to Chapter 11 of the Castro Marine Biology textbook, which covers the intertidal zone. The intertidal zone, some of you know as the tide pools, but the intertidal zone is the area between the low and the high tides. That's the mean high tide and the mean low tide. So on the diagram right here, it would be this area right here is what we're concentrating on today. That's the intertidal zone. In another talk, we'll take a look at the subtidal zone. Subtidal zone is below the low tide and has a different mix of organisms. We'll take a look at that when we cover the estuaries, and we'll see that the salt marshes and the mangroves are found in the subtidal zone. Today, though, the intertidal zone, the thing about the intertidal zone is organisms that live here have to be adapted to all kinds of different situations, especially the fact that for part of the day, they're going to be out of the water, which will increase the temperature, and they've got to stay moist. So let's see how those guys do that. Well, before we do that, let's take a look at the substrates. Remember, substrate is what the organisms can latch onto at the bottom. And so in the intertidal zone, you have different types of substrates. You have a rocky substrate. So sometimes we call the tide pools the rocky shores. So the rocky substrate can vary as to the type of rock and the slope angle. Okay, where I live, the tide pools drop off pretty quickly because there's no continental shelf, and the mountains run right up to the coast. And so you have some very large rocks coming off the continents, and those line the rocky shore regions. Over here, picture on the right shows you some sandy beaches, which can be intertidal areas. Remember, just if it's between the high tide and low tide, and the soft bottom can vary from sand to silt, or a mixture, which is mud. That silt is really packed. We're going to see, and so different organisms live in different types of soft bottoms. In fact, let's take a look at now the organisms that live just on the top of the substrate. Those are called the epifauna, and the ones that live inside of the substrate, that's the infauna. So on top is the epi, and inside is the infauna. Now, we also have something called the meiofauna. And the meiofauna are organisms not even like this one, but they're so small that they're microscopic, and they can go between the grains of sand, if you will. Okay, so some of these move around even within the sand. A lot of them are just stuck there. So I want to remind you of the term sessile. Sessile organisms are organisms that don't move around, but some of these meiofauna do have the ability to move within that substrate, even things like silt, which is, when you think of it, pretty crazy. All right, so if you live in an intertidal like this sea star does, well, you have all kinds of problems that you've got to cope with, like, for example, desiccation, high temperatures, salinity. These are some of the main ones there. Interrupted feeding, because if you're a filter feeder, you've got to be in the water, and when you're out of water, then you aren't feeding. You've got to be adapted to be able to stay in place when you have the power of the waves hitting you. Oxygen availability, buildup of carbon dioxide at low tide, because you have pools of water. Those pools of water can be influenced by whatever you have there. You might have a bunch of algae that are living in there. You might have too many organisms in one spot that would create too much carbon dioxide. And by the time the water comes out, you might have limited spaces that have water in them. So the tide pools are going to be places where there's going to be a lot of challenges for organisms to meet when they are living in the intertidal. Desiccation means that they dried out. And so when exposed organisms must deal with potential water loss. Water loss is more pronounced on hot, dry, or we forget, also windy days. The wind increases the evaporation of the water, and organisms can deal with the potential water loss by hiding or clamming up. There's a we'll see that on the next slide, but here's the reindeer clam in Lake Pontchartrain in Louisiana. And so it's an example of a intertidal organism, and you can see that it is, in this case, all clammed up. So desiccation is something you've got to be able to cope with. Also, what you want to do is maybe you're hiding. As your hiding may involve moving to a tide pool or an area with more moisture. So throughout the day, as the low tide goes down and there are pools of water, then you might have organisms like these chitons that are trying to find an isolated pool of water. So this is true for organisms that can move around. Remember, the term for that is modal. Some organisms are going to go to different areas says you're hiding may also mean that some organisms only live in areas where moisture will remain when the tide is out. So they seek out these areas through experience. They know that this particular spot on this rock, this particular crevice is not going to dry out. It might be that there's a dip in the rocks that causes a small deformation in the stone in the rock, and so it forms a pond that has enough water that lasts throughout the cycle and such. So yeah, some of these guys will move to places that have water. Other organisms will just close up. And so I already showed you the clams from Lake Pontchartrain in Louisiana. Near me, I have a photo of some mussel that are at Point Fermin, which is near Long Beach, California. If you were to go to the Cabrera Marine Museum and go out the back door, leads right to Point Fermin right there. So organisms can deal with the potential water loss by clamming up. Obviously, that means closing the shell or otherwise walling itself off from the government or from the environment in an attempt to conserve moisture, such as an oyster closing its shell or a snail walling itself off using its operculum. Operculum is the cover. Like if you've ever seen a snail, I don't know, in my youth, we would take snails and put salt on them, and then they would start to bubble and the snail would go back into its shell. Well, there's a little, oblong, round shell at the end of the fleshy part of the snail that it uses like the door to the shell. That's called the operculum. And so close it up so that you don't lose any water. It says here, while moisture can be conserved this way, there is a downside. There's no gas exchange and there's no feeding. So you're going to have to, when situations get better, you have to open it back up and get what you can while you can. Alrighty, so that's the way that organisms take care of desiccation. Temperature is also a problem in the intertidal zone. We have a wide range of temperatures that must be tolerated by organisms in the intertidal. This can be wide ranging. They can, imagine a very hot day. The ocean temperatures, in the 50s, really lucky, might be lower than that. And on a hot day, the external temperature, the ambient atmospheric temperature might be 90, maybe even 100, depends where you are. And so these organisms that are intertidal have to have certain mechanisms to stay cool. Like, for example, here is a Nerita placata, which is a type of snail, and you can see that it is white, which reflects the sunlight, and then you have ridges which act as surfaces where you could have some water evaporation, and that evaporation by cooling will help that organism stay cool. Another problem is the salt. And so if you're an intertidal organism, you know, the ocean is what, 35 parts per thousand salt, you know, mostly sodium chloride, some other ions as well. Organisms in intertidal are normally urahaline, and that means they can go and they have a tolerance to different salinities, whether it's really salty or even almost like the fresh water coming out. Now, fresh water is not going to be a 0% salinity or anything, but it's going to have a reduced amount of salt in it. So it might be 1 to 5, depends on where you are. Okay. And so most organisms are urahaline, and the urahaline organisms can tolerate changes in salinity. But subtidal organisms are normally stenohaline. That means they don't tolerate big changes in the salinity. They tolerate very low variations in salinity, and that's why they stay in the water all the time. Okay. Also, that's why you don't find these organisms as frequently in estuary regions, because in estuaries, that's where the fresh water and the salt water mix, and then you have a various salinity value. So instead of 35, it might be 30, it might be 20, depending on where you are. Right, and I think I've already said some of this salinity may begin at 20, 25 parts per thousand and climb dramatically during the day due to water loss when the intertidal is exposed. Okay. Salinity may also drop during the day due to the sudden influx of fresh water provided by the passing thunderstorm. That's an isolated event, but certainly could happen. And a salinity change of possibly 20 parts per thousand or more, combined with a temperature change of 40 degrees or more, may occur. So these are wide ranges. These are big changes in both temperature and salinity. So organisms that are intertidal organisms need to be adapted to both of those different types of changes. All righty. Interrupted feeding, I mentioned earlier. It happens for filter feeders. Few organisms, such as snails, can still feed during low tide by scraping algae off surfaces. That's how they typically eat, but when they do that, there's the higher chance that they could dry out. For communities with semi-diurnal tides, an animal would spend nearly half of their day exposed and not feeding. So they're going to have some type of physiological mechanism to cope with that. All right, let's take a look at waves in the intertidal. Wave exposure can be a problem for intertidal organisms. Rocky shores are often exposed to significant wave action. So you're going to see waves crashing on the rocks. Some of the more classic things, when you go to a rocky shore region, some will even come up and look like a blowhole. There's one in Ensenada called La Bufadora, and when the waves come in, makes a big splash. And so imagine being subjected to that all the time. Well, how do these intertidal organisms, how do they do that? Well, look at this one. There's a sea anemone that's able to crouch down, if you will, when there's a heavy wave action. There are mussels and mussels are clam-like organisms, and those are going to have a special thread. They're called byssal threads, and those byssal threads allow the mussel to attach to a substrate. Hopefully it's rocky. Here's another one. This guy is standing up straight, but when the wind comes, it's able to bend and minimize the effects of the wave on the organism itself. And so different ways to do it. You can crouch down, you can just go with the flow, or you can stick really well. All right. Oxygen availability. Oxygen can be exhausted if an organism clams up during low tide. You're trying to deal with the lack of water, not dry out. And if you're closing up, then you've got to either store oxygen or have some way to get some oxygen. It says here it can also be exhausted in tide pools if there's a high density of organisms at night. Says here particularly algae at night. Algae go through photosynthesis, as you know, and what they do is they take up the carbon dioxide and as they take up the carbon dioxide, a waste product of photosynthesis is oxygen. So if they're not going through photosynthesis, then they're not putting out the oxygen, so that oxygen could get used up. Also, since you're not going through photosynthesis, the carbon dioxide could also build up because it's not being taken out of that very small area. It's like having a fish tank. I'm looking at the fish tanks in my laboratory. Some aquarists will tell you that, well, I like to have a bigger fish tank, like a 60 or 100-gallon fish tank, rather than I have 7 10-gallon fish tanks that I'm currently running. Why is that? It's better to have a big tank than several small tanks. Well, some people would say that the bigger tanks are more forgiving as far as, when you have as far as the temperature changes, for example, there aren't going to be wide temperature changes in 100 gallons of water rather than 70 gallons in 10 different tanks. It could be a wide range of temperature. And so it's easier to take care of the larger bodies of water, even if they're just bodies of water within the tide pools. And so those, again, a larger body of water might be more forgiving. That's why we can dump trash and chemicals into the ocean. It seems like nothing's happening because the ocean's so huge. But as we know, that's starting to become a problem. There are parts of the ocean that are basically devoid of any fish that are usable for people. There are parts of the ocean that are accumulating plastic garbage. So yeah, larger bodies of water are more forgiving, but only up to an extent. Back to the tide pools, though. There's limited space in tide pools, and that's another problem along with desiccation and temperature and interrupted feeding. In some intertidal communities, space may be limited. This is especially particularly true in the rocky intertidal, where surface area is limited. But this can also happen in soft body communities, especially when you have things like oysters and clams that are trying to stick to something that has more substance to it than sand. And they're doing that so that they can stay in one place. And eventually what happens when you have a shoal is that they start sticking to each other, and then you have like it says there, an oyster reef or a mussel shoal. Alrighty, so limited space in the intertidal. Also, zonation in the intertidal. I want to say a couple things about that. If you take a look at the picture, there are differences even within the intertidal zone. You have the upper, the middle, and the lower, and you have different organisms that live in different parts. That's because you're going to have more exposure in the upper intertidal to the air and to a higher temperature. Mostly the lower intertidal is going to be underwater most of the time and relatively longer than the upper intertidal. So you can see a different mix of organisms that you would expect in all these different layers. Competition would be greater in a lower intertidal because it is the least extreme of the zones. That means there's more organisms that can live in the lower tidal because of the abiotic conditions. There are fewer relative organisms that are adapted to living in the upper intertidal. And then the lower intertidal always has more species for the same reason, and that makes a lot of sense. Competition. Some species are better competitors than others and will exclude others if the community is left undisturbed. Remember, there's the biological idea of Gauss's law of competitive exclusion. And what that says basically is that organisms, when they're competing, will either win or they'll lose and die off, or they'll lose and move away. So those are the outcomes of competition. So there's going to be more competition down lower in the intertidal zone. If a habitat is disturbed, organisms move in or excluded in a predictable pattern known as ecological succession, something we covered in a different lecture. But basically, if you have some organisms that are good colonizers, we call those pioneer species. And as they change the environment ever so slightly, other flora and fauna are able to now live in this community, and different plants and animals are going to replace the original ones. And finally, you'll have an end result called the climax community. And that forms when the community is undisturbed for a long period of time with similar physical conditions. And then if you have an area with similar physical conditions like temperature, desiccation, like the ones that we mentioned earlier, and you have that competition, then you're going to have an expectation of certain plants and then animals that are attracted by the plants to that climax community. Things like, the kelp forests have a particular mix of organisms that you would expect. The coral reefs have a particular mix of organisms that you would expect. And here, the rocky shores, the intertidal zone, has a particular type of plants and animals that you would expect with all the different physical characteristics of the area. The keystone species I mentioned earlier, this talks about keystone predators, such as sea stars that strongly affect the currents and density of animals in the middle intertidal. So if you take a look at the picture right here, what's happening is that when you take out the sea stars artificially, like if we were collecting sea stars, and when you take them out of the local ecosystem, then what happens is that the mussels here are going to take over, because the mussels are not in check anymore, and the starfish, what they do is they take a mussel and they open. They can, they have their water vascular system, and they have the ability to put their ampullae right on the shells, and then with that water vascular system, it's like a hydraulic system, and it pulls the shell apart. And even if you have a small opening, say 2-3 millimeters, then what can happen is the stomach can then evert itself and go out of the starfish and go into the shellfish, and then it'll just sit there for a long time, and it'll just degrade the muscle from the inside there. It's like the stomach is sitting inside of the shell. And so when it's all done, then the starfish, the stomach goes right back through the shell and back into the sea star, and then it does it all over again. Well, if you take those sea stars out of the environment, then that's going to be a problem. So that's why sea stars in this particular example are examples of keystone predators. In the kelp forest I mentioned, and we'll see in more detail in the kelp forest talk, the sea otters actually keep the sea stars in check. And so sea stars, sea urchins, they will be kept in check by the sea otters. So the sea otters are known in the kelp forest as a keystone species. All righty. Here's another slide on zonation. It says biological interactions are often important factors in determining lower limits of rocky intertidal organisms, physical factors in determining the upper limits. Okay, so back to this picture right over here. That's what this is talking about. The upper limit is determined by the physical factors of the tides coming out, and then down here it just depends on competition between the species that are living right there in that particular part of the world. Right, and so this gives you another idea. This example, this is a classic example of barnacles. Were studied at the University of Washington. You can see there are two different species of barnacles here. These are called the little gray barnacles, and here are the rock barnacles down here. And so we have typically some predation happening down here at the bottom, but at the top, the limiting factor is that they're drying out. So this is the desiccation that occurs right here. And so this is the practical limit. This diagram here shows you the realized niche, where over here it's more of a theoretical niche or area where these guys might possibly live. But right here, this rock barnacle can live through this whole range, but practically speaking, it doesn't. And the reason that it doesn't is because down here, it's competing with other organisms, and up here, they're getting dried out, and these guys are better at living in a environment that has come down from the high tide. So these guys are better at dealing with desiccation due to the retreat of the water during the daily tidal patterns. So this is again called a realized niche, and over here this is the theoretical niche. This is where these guys live. And even if you look at the little gray barnacles over here, and by the way, this is the story of Balanus and Thalanus are the different species. And so these are the common names of the different species: little gray barnacles, rock barnacles. But you can see that this range is much smaller than the range where if it had no competition and it had no limiting factors, then it could stay and live on this whole part right there. All righty. Earlier I mentioned the sedimentation, and they're in areas that have a lot of gravel or sand. You might have neofauna that live in between the sand grains here. Now, in silt and clay, look how compacted these are. And so you're going to have, different organisms, microscopic organisms that can live within these sand particles here, or these clay and silt particles. But over here with the sand, you have more space between the sand particles because of the irregularities of the surface, and even more so when you have gravel. So you also have, if it in the smaller size sediments here, you're going to have less oxygen because if the water comes in and can go through, and the water carries oxygen along with it. So that's some sedimentation. And so there are different organisms that can live in these different types of soil because of the size and because of the amount of oxygen that can go through. Remember, with really small things like silt, you're going to have, it's going to be more compacted and it's going to have less water going through. So it has less oxygen going through. Okay, so sediment size can be small so that all except the top few inches is anoxic. Remember, A is a prefix that means without. So without oxygen, this environment is anoxic. The infauna, remember, are the ones that live inside the sand or silt or mud, and says that it maintains a connection with the oxygenated zone via tubes, siphons, or extended burrows if they're located in the anoxic zone. There's also at the bacteria level, there's anaerobic bacteria that break down this organic matter that's in the anoxic zone. This is why sometimes, in different areas, you're going to have a stench associated with the anaerobic breakdown of the organic material. You might get a really stinky sulfur smell to it. Most animals are in fauna with adaptations to dig or burrow. So the organism can go in to the sediments and dig their way in. The epifauna, remember, the ones that live just on the top there, but they're poor because they are more susceptible to whatever's out there, like competition, like predation, more exposed to the environmental cues. So more adapted to, a better adaptation is to be an infauna. And so here's the soft-bodied intertidal zone, not the rocky shores down, but the soft-bodied, just to give you an example of different organisms that live in this area. Here's the low tide right there. So these are organisms that are subtidal. But over here, you're going to have organisms that are intertidal. You can see, look at the top surface, there's nothing there. Why is that? Because all these guys are digging down when you need to. Okay, and then this gives you an idea of the food webs in the intertidal zone for both the soft-bodied animals and also soft-bottom organisms and all environments, and also the rocky shore environment right there. So looking at the food web for the soft bottoms, you can see here that the primary producers are the algae and the seaweed right there. And then you have organisms that feed off of that. And these guys right here are the producers of the whole soft bottom intertidal zone. All right, and then here is, if you will, the trophic level diagram for a rocky shore food web. You can see at the very bottom, you have seaweeds, you have golden algae, the diatoms, seagrasses, which are usually higher plants. So these guys are the ones, this is where photosynthesis happens, and the organisms that graze off of that are the herbivores. And then over here we have carnivores that eat the herbivores, the scavengers that go and pick up all the organic waste. And then over here you have filter feeders that are taking the microscopic stuff out of the water that's still organic. And amazing that they have enough food to eat, but they filter an amazing amount of water through through even one clam or mussel in a minute. So pretty crazy. Some sponges, speaking of filter feeders, can pump a gallon of water every minute through their bodies and filter out the microscopic organic materials that serve as their foods. Alrighty. So those are some comments about the rocky shores today. Hope you learned something new. And we'll see you next time. I'm Mr. Vallejo. Have a great day.